fluids from one site to another. Yet, despite the importance of tubes, their detailed molecular understanding of how tubes regulate their size and shape remains unclear. So, in our lab, a fascinating type of tube that we look at are single-celled seamless tubes. These are actually the smallest types of tubes that have been described, and they get their name because they form intercellularly and they lack any observable junctions. They can be found in the example shown here, the fly tracheal system, where terminal cells form seamless tubes, along with the vertebrate vasculature, as shown here in intersegmental vessels in zebrafish. And in our favorite model system, the C. elegans excretory canal, which is this fascinating H-shaped cell that spans the entire length of the adult worm and forms and expands its lumen intracellularly. So what we're really interested in is what is actually coordinating this type of lumen expansion, using this as a model system. One clue as to how this may occur comes from an EM analysis that was conducted in our lab, that was also appreciated previously by the Gobel and Labo S labs, where you can appreciate these large canalicular vesicles that are filling the cytoplasm of the excretory canal. If you look a little more closely, you can see that several of these vesicles are connected directly to the luminal membrane, but they're never observed to be connected to the outer basal membrane. So the hypothesis that had been put forth is that these vesicles are indeed contributing membrane somehow during lumen expansion, and they're required for the growth of this seamless tube. So we wondered if that was indeed the case, and if so, how are the vesicles being targeted to the luminal surface? So that led us to think about the exocyst complex because of its conserved role across species in regulating polarized membrane trafficking. So I'm showing you the complex here in purple. It's composed of 8 proteins, and there's a GTPase called RAL1 that's required for its activity. So for the purposes of my talk today, I'll be focusing on 2 core components, Sec10 and Sec5, along with RAL1. So first we wanted to ask if the exocyst was actually expressed in the canal side. So we endogenously tagged Sec5 with YFP, and you can see that it nicely labels the luminal domain of the excretory canal. And in the figures I'll show you today, the dotted line represents the cytoplasm of this zoomed-in image of the canal. So this, along with the previous observation that RNAi knockdown of exocyst in MDCK cells prevents multicellular lumen formation, led us to ask if the exocyst is actually indeed required for seamless tube growth. So that will be the first part of the talk, and then I am going to talk about how the exocyst actually gets recruited to the luminal domain, and finally talk a little more broadly about some molecular mechanisms that could be involved in polarizing seamless tubes. So for the first part, to look at exocysts, we actually needed a strategy to deplete the protein in a tissue-specific manner because mutants are embryonic lethal. So we turned to the ZF1 degron method used in C. elegans, where we can take a small ZF1 tag to add to a target protein, and then re-express the ZIF1 adapter using either a heat shock or tissue-specific promoter to recruit ubiquitination machinery to the target protein and rapidly degrade it. So we tested this using a transgene in our lab, where we've expressed ZF1 GFP in all tissues, including the excretory canal. And then we expressed ZIF1 with a canal-specific promoter, and we can show that it very nicely degrades the GFP signal just in the excretory canal. So using this strategy, we then wanted to go in and remove components of the exocyst and then look at the morphology of the canal using a cytoplasmic and a luminal marker. So in wild type, you can see there's this continuous lumen shown in cyan, and the cytoplasm is more broadly expressed in yellow. But when we take out Sec5 or RAL1, you can appreciate these large cyst-like structures that occur in the animals. To quantify this, we decided to measure the total growth of the canal as a percentage of body length. So in a wild type, you can see almost all the animals grow the canal to the end of the body at L4 stage. If we take out Sec5 or RAL, you can appreciate that this is severely truncated. So this suggests that RAL and exocysts are indeed required in the excretory cell. So for the next part of the talk, I want to talk about ways in which the exocyst could actually be directed to the luminal membrane in order to perform its function. So Nelio gave a really nice introduction on the PAR complex, and I'll just briefly point out some of the players I'll be referring to. So the reason we decided to look at the PAR complex, I should say, is because of its highly conserved role in cell polarity in many different organisms and cellular contexts, and we thought maybe in the canal cell it was also key for polarizing the tissue. So PAR3 and PAR6 are the PDZ domain scaffolding proteins. APKC is the kinase. And then CDC42 is a small GTPase that, through the work of the Gehring lab, along with others, was recently appreciated to bind in the early C. elegans embryo to PAR6 to maintain it in a more active state than the PAR3 bound form. In addition, some other reports looking at PAR components in the fly trachea suggest that they're indeed required for the localization of exocyst. And more recently in mammalian cell culture, an interaction between PAR3 and exocyst was observed to recruit the exocyst to tight junctions. So we wanted to look in the canal cell to see if PAR proteins were indeed present and polarized. So we used CRISPR to tag PAR6, PKC3, and PAR3. And you can see right away that just like the exocyst, they nicely localize the luminal domain. CDC42 actually showed us a broader distribution. So compared here to PAR6, you can appreciate that CDC42 is expressed within a region of the cytoplasm as well as at the luminal domain. And in this line trace, you can perhaps appreciate that even better, that PAR6 tightly localizes to the lumen, whereas CDC42 is more broadly expressed. So what actually happens when you perturb PAR function in the canal? So using the same method as I showed with the exocyst, we took out PAR6, APKC, and CDC42, and saw a similar phenotype where there are these truncated cystic canals. With PAR3, interestingly, the canals actually looked wild type at this stage. And you can appreciate that using the length quantification. So, like I mentioned, for PKC3, PAR6, CDC42, there's a truncation defect, but PAR3 resembles the control. So I should mention that if you look early at PAR3 depleted canals, there is actually a defect that appears to resolve over time. So we think that we're still investigating it, but we think PAR3 might be acting earlier during polarization compared to the later maintenance phase. So to actually test the epistasis between exocyst and PAR components, we had to modify our degradation to actually remove PAR proteins after the canal is fully formed to get around these morphological defects. So for this, we used a heat shock promoter to drive ZIF1. And here I'm showing you a PAR6 endogenously tagged with ZF1 and YFP, which is brightly expressed in the canal. But when we heat shock to activate ZIF1 and look about 45 minutes later, we see that it's very robustly degraded from the canal cell. So using this, we can then go back and look at the localization of exocyst components in the loss of PAR6. So for this experiment, I'm using PAR6 again with the ZF1 tag and looking at Sec10 in the red channel here, which is nicely localized to the lumen, just like PAR6. When you activate ZIF1 to remove PAR6, you actually see that there's a change in the distribution. So now it becomes more cytoplasmic, which you can appreciate in this trace, along with the quantification I'm showing here, which is a ratio of the luminal to the cytoplasmic intensity of the Sec10 signal. So you can see that that's going down after loss of PAR6. In contrast, when we took out PAR3, we actually didn't see any change in the exocyst distribution. So you can see here it looks very similar to the wild type, and in this quantification as well. So this data, along with our phenotypic data on PAR3, suggests that PAR6 and APKC are actually the more active arm of the PAR complex in recruiting the exocyst, whereas PAR3 doesn't appear to be playing a role at this stage. So for the final part of the talk, I just wanted to talk about what mechanisms we think could be acting upstream of PAR6 and APKC to position them at the luminal membrane to enable the polarization of the exocyst. So I mentioned before that a known activator of PAR6 and APKC is the GTPase CDC42. And previous work in the C. elegans canal has implicated a CDC42 ROGEF called EXC5 or FGD in mammals, which you can see here in this transgene is expressed in the canal, and in a mutant allele, there is a cystic phenotype which looks similar to PAR6 or CDC42. Also, in data I don't have time to show, I did used the ZIF1 heat shock degradation to remove CDC42, and I saw a reduction in PAR6 at the luminal membrane, suggesting that CDC42 was indeed recruiting PAR6 in this system. But we decided to look more closely at EXC5, so we tagged it with ZF1 and M scarlet, and you can see it nicely patterns the luminal domain. And in this experiment, I'm going to be looking at PAR6's binding partner, APKC, which, similar to PAR6, is brightly expressed at the lumen. When we use heat shock to remove EXC5, we now actually see a severe reduction in PKC3 labeling of the lumen, which you can appreciate with these quantifications as well. So that brings me to the final model, which hopefully I've convinced you of some today, but it's still currently being worked on. That EXC5 is responsible for activating CDC42 at the luminal membrane. This enables the recruitment of PAR6 APKC complex and brings the exocyst to the membrane where vesicles can efficiently be trafficked to expand the luminal domain. Now, just to circle back to the previous results on PAR3, we've shown that it's not required for exocyst localization during lumen maintenance at that later stage, but it could have a role either earlier in lumen development, or this could represent an example with PAR complex interfacing with exocyst or other components differently depending on the tissue or the context with which you're looking, which has been reported before. So finally, I'd like to thank all the members of the NANCE lab, especially my mentor, Jeremy, ChemFuse, Dickinson, and Goldstein labs for CRISPR alleles used in this study, funding sources, and other members of the Scribble Institute for helpful discussions. And I'll happy to take any questions now or by email. So thank you.
Great, thank you so much. That was great. So we have some questions from the Q&A. Sorry, that's the delayed clapping. We have some questions from the Q&A. Are the vesicles Golgi derived, and do you know if they recycle from the plasma membrane or the luminal membrane?
That's a good question. So the canalicular vesicles are they are thought to be Golgi-derived vesicles, and in the EM slices, yeah, I mean, it's hard to appreciate the vesicles that are tethered to the lumen, whether they're fusing or budding off the lumen. But because the exocyst is expressed at the lumen, we know at least, from what we know about the exocyst, at least a population of them are being recruited to the luminal domain. But something we've been trying to do in the lab is get tools going to do more live vesicle imaging to try and answer those types of questions.
Great, thank you. And I want to say there's been some questions about whether people are